presentation.
So thank you for a nice introduction, and also thank you for inviting me to this interesting seminar series. I would just like to confirm that you can see my screen now with the presentation. Yeah. Okay. Thank you. Today I would like to present our recent work, which we published in Science Immunology, and which was focused on the restoring tumor immunogenicity with dendritic cell reprogramming of cancer cells. As we all know, in the past years, the cancer immunotherapy basically changed the way how cancer is being treated, and development of multiple immunotherapeutic strategies, and especially checkpoint inhibitors. I'll just use my pointer. Checkpoint inhibitors in particle brought a lot of change to the way how cancer is being treated nowadays and prospectively in the future. Using immunotherapy, it suddenly became possible to achieve long-term and durable remissions, even in patients who were previously very hard to treat, like those with advanced or metastatic melanoma. However, the major challenge standing for the immunotherapy is actually that most of the patients kind of don't respond to the therapy or later fail on the therapy. Obviously, for the efficiency of the anti-cancer immunity, and also the efficiency of the responses to immunotherapy, there are two important aspects which needs to be fulfilled, and it's the antigen presentation by cancer cells, and also the expression of the cancer antigens, and also the presentation of those antigens by dendritic cells to T cells, which then leads to the T cell expansion. And currently, it's becoming more and more apparent that in this anti-tumor circle, there's one cell type which has exceptional functions, and these are so-called CDC1s, or type 1 dendritic cells. Type 1 dendritic cells are excellent in the cross-presentation of antigens and presentation them to the T cells, which leads to their active expansion. But they can also activate other effector immune cell types, including NK cells. And basically, they are helping to feed this whole anti-cancer immunity loop. Recent papers also showed that the presence of the CDC1s within the tumor microenvironment was then associated with better outcome of patients, and also with better responses to the immunotherapy. But as you can expect, this population is very rare, and especially in the context of the cancer patients, CDC1 cells are frequently functionally compromised or being excluded from the tumor microenvironment. However, due to modern science advances, we have now different ways how to actually generate de novo, like even very rare cell subtypes. Well, already for quite some time, we know that the identity of the cells is actually not irreversible, and basically every cell in our body can be changed. We can revert all the cells, like either back to pluripotency using iPSC reprogramming, or we can take one terminally differentiated cell and convert it to another unrelated terminally differentiated cell type using a process called direct cell reprogramming. And basically, the only thing you need to know to do this is to identify a specific combination of transcription factors, which are then responsible for driving the new cellular identity. So far, cellular reprogramming was used mainly in the context of regenerative medicine and cell replacement therapy, and it helped to generate multiple cellular types, including different types of neurons, also functional cardiomyocytes, or, for example, also functional hepatocytes. And in our lab, we actually wanted to bring the cellular reprogramming more to the context of cancer immunotherapy. Some years ago, we identified a combination of three transcription factors, PU1, IRF8, and BAT3, which are actually able to initiate the CDC1 or type 1 dendritic cell identity in mouse fibroblasts. And later, we also described how these three transcription factors cooperate to silence the fibroblast genes and induce the CDC1 transcriptomic program in the fibroblasts. And in the current project, we took this concept a step further, and we aim to use actually and overexpress these CDC1 master regulating transcription factors directly in tumor cells. By this way, we would actually generate very interesting cell type from the original cancer cells, so-called tumor antigen presenting cells, or as we abbreviated, tumor APCs. These reprogrammed cancer cells will obviously have several advantages, because we will not only force them to show again their present their cancer antigens, but given the fact that they will be similar to CDC1s, they will be actually able to directly activate and show the antigens and directly activate and clonally expand antigen-specific T cells, and also will be able to crosstalk with other immune effectors, including NK cells, and by that way should be able to reinitiate or re-engage the anti-tumor immune responses and lead to the eradication of the cancer. To begin with, we wanted to test our hypothesis, and we gathered a panel of human and mouse cancer cell lines and primary cells, and we wanted to know how these cells are being actually changed during the reprogramming process, like what's happening with their transcriptome, epigenome, how their phenotype is changing, and also like if they are really gaining functions which are typical for CDC1 cells. In the mouse system, we started with reprogramming two poorly immunogenic cancer cell lines. One was LLC, this is derived from mouse lung carcinoma, and B16, which is derived from mouse melanoma. And as the markers of reprogramming to the CDC1 lineage, we selected the simultaneous expression of CD45, which marks the transition to the hematopoietic lineage, and the expression of MHC2 as marker for antigen presentation, because based on our previous publications, we knew that these cells are actually reflecting well the reprogramming trajectory towards CDC1. And you can see that we managed to induce those reprogrammed cells in both mouse cancer cell lines. Next, we wanted to evaluate the CDC1 reprogramming in a wide range of human cancer cells derived from multiple malignancies all over the body. And you can see on the graph that we basically managed to induce the reprogramming or triggered reprogramming and induce the emergence of CD45 positive, HLA-DR positive reprogrammed cells in all malignancies or all cell lines, but with different efficiency, which varied according to the origin of the cancer. And what was interesting, we also detected quite large proportions of cells which were only partially reprogrammed. These you can see like in the blue and green, and those cells expressed only one of the reprogramming markers. Together, we also confirmed that the reprogrammed cells expressed other CDC1 specific markers, including CLEC9A, CD226, or CD11C, and that they significantly changed morphology, because here is cell before reprogramming, here after reprogramming, and I know the picture is small, but should be visible how the cells are creating like those dendrites and really resembling real dendritic cells. We also wanted to look a little bit deeper into the cell using transcriptomic analysis. And when we did this, we saw that the reprogrammed cells, and also both partially and completely, which are shown in triangles and squares, actually moved from their non-reprogrammed counterparts quite a lot and very close towards the natural CDC1 population. But of course, not all of them reprogrammed exactly the same way or to the same extent. So to be able to address the variability of the reprogramming, we also generated a signature of genes which are commonly expressed in the CDC1s. And then we compared our reprogrammed cells like against this signature. And we saw that despite the variability of reprogramming which we saw, all of the cell lines, all of the reprogrammed cells, either partially or completely reprogrammed, scored above 50% of this signature, and also enriched in terms which were associated with antigen presentation or other immune-related processes. During our initial experiments, we also noticed that reprogramming to the CDC1 is actually a very fast process. First, reprogrammed populations we are able to detect already at day 3 of the reprogramming, and then this population is just becoming bigger towards the later reprogramming time points. So to be able to say something more about the dynamics of the process, we sorted these partially and completely reprogrammed populations along the time course, and then we subjected them to the analysis with ADAC sequencing and mRNA sequencing. On the transcriptomic level, you can see that the CDC1 transcriptional program was acquired stepwise or progressively towards the later time points. All the stages kind of clustered together and very far from the parental population. And then the day 7, day 9, we're getting very close to the natural CDC1 population. This fast transcriptomic rewiring was probably enabled by the very rapid epigenetic remodeling, which we saw due to the data from ATAC sequencing, where you see that the major change must be happening already before the day 3, because the cells from day 3 to day 9, they basically cluster all together and very close to the CDC1 population, which means that the reprogramming is initiated very early after the transduction, and the later stages of the reprogramming probably are associated more with maturation or the stabilization of the newly acquired identity. Using transcriptomic analysis, we also showed that the reprogrammed tumor APCs progressively activated genes which were associated with antigen presentation and processing. And at day 9, they were reaching similar levels as we observed within the normal CDC1 cells, which supports the idea that they will be able to present tumor antigens. However, to be able to really say that tumor APCs can truly present cancer antigens, we decided to perform immunopeptidomic analysis in mouse melanoma cell line. And we saw that the cells after reprogramming actually enhanced expression of those peptides. Compared to the non-reprogrammed cells and also to the positive control, which was cells treated with interferon gamma, which also results in overexpression of MHC1. Interestingly, among those peptides which we identified, there were also multiple known tumor antigens associated with melanoma. And the fact that we were able to detect them was especially interesting in the light of the fact that on the transcriptomic analysis we saw that the genes which are encoding those tumor antigens are actually being mostly substantially downregulated following the reprogramming process, which shows that the enhanced activation of the antigen presentation in the tumor APCs basically outweighs the downregulation of the tumor antigens following the reprogramming into tumor APCs. Importantly, reprogrammed cells also induced expression of costimulatory molecules, including CD40, CD80, or CD86. And this is very important because costimulatory molecules serve as the second signal which is necessary for engagement and productive activation of T cells. And to further support the increased immunogenicity and ongoing antigen presentation in the tumor APCs, we also showed that cancer cell lines which were expressing ovalbumin were more prone to get killed by T cells specific against ovalbumin, as you can see here. And further, we validated this killing or this increased immunogenicity also using melanoma cells when they were co-cultured with cytotoxic T cells specific against PMEL, which is one of the common endogenously expressed melanoma antigens. And you see that also those cells were increasingly susceptible to the killing by PMEL specific T cells, which supports that tumor APCs can really present and cross-present tumor antigens. Altogether, we showed that tumor APCs are activating antigen presentation and that they are becoming very immunogenic. But we also wanted to investigate, because this is reprogramming into CDC1s, whether they acquire other functions typical for natural type 1 dendritic cells. To this end, we showed that reprogrammed cancer cells responded to TLR stimulation, and then they secreted pro-inflammatory cytokines, including IL-12, IL-29, CXCL10, or TNF-alpha, which then further contribute to the global anti-cancer immune responses. Furthermore, we also demonstrated that the reprogrammed cells are able to engulf small proteins and particles from the media, as here is documented by the uptake of fluorescently labeled ovalbumin, here like in red or on the pictures, and that they can also process those antigens, as here we document by the detection of cleaved DQ-OVA in the reprogrammed cells. Another hallmark ability of CDC1 cells is undeniably also the capacity to phagocyte dead cells. And as you can see on this video, here's like one green reprogrammed cells, which is actively scavenging and looking for and engulfing fluorescently labeled dead cells, which proves that tumor APCs are truly endowed with the capacity to phagocyte. And finally, we also address the ability of tumor APCs to cross-present antigens. And here, when we pulsed B16 melanoma cells with OVA before and after reprogramming, we noticed that the reprogrammed cells are actually able to induce induce proliferation and also activation of antigen-specific CD8 positive T cells, which proves that they are really able of the capacity to cross-present antigens to cytotoxic T cells. Considering future clinical translation of our approach, we also understood that it's very crucial to address whether CDC1 reprogramming can be elicited in primary human tumor tissues. And to prove this, we gathered a panel of diverse primary cancer samples from multiple cancer types. And we showed that basically the reprogramming can be induced in all those samples in a similar extent as we observed before with the panel of cancer cell lines. Using single-cell RNA sequencing, we then also validated that despite the different origin of the cancer cells after reprogramming, here in red, they are starting to cluster together and close to the natural CDC1 population, which is shown as this little gray tip. The reprogrammed cells also upregulated expression of typical CDC1 markers, and importantly, they also launched endogenous expression of IRF8 and BAT3, which shows that the adoption of the new identity is rather stable and not dependent on the continuous expression of the exogenous reprogramming factors. Importantly, following the reprogramming, the cells will affiliated with high fidelity and high frequencies exclusively to the CDC1 subset after reprogramming, and not to the other DC subsets, which supports that our reprogramming strategy with PU1, IRF8, and BAT3 is very specific to generate only CDC1 cells. To address also the functional capacity of tumor APCs, which are derived from primary cancer cells and not only from cancer cell lines, we used the opportunity of the primary melanoma cells and demonstrated that when we reprogram them and stimulate them with long MART1 peptide, they are actually able to activate and expand the population of MART1 specific T cells, which further supports the antigen presentation. And importantly, also when we co-cultured the primary melanoma cells after reprogramming with autologous tumor infiltrating lymphocytes, it also led to the activation of the tails. And then the activation was translated into increased cytolytic reaction against the reprogrammed cells compared to the non-reprogrammed counterparts, which is showing or giving another evidence for efficient presentation of endogenous neoantigens. And since we are talking, or I'm talking all the time about reprogramming of cancer cells, it's also important to mention that the adoption of CDC1 identity was accompanied with loss of tumorogenicity. Here you see in vitro the soft agar assay, where the reprogrammed cells were no longer able to generate colonies, unlike the non-reprogrammed, only mock transduced cells. And also in the context of in vivo, they were no longer, after reprogramming, able to induce tumors when injected into the animals. Because all the animals which were injected with reprogrammed cells, they stayed tumor-free for the whole duration of the experiment, while the control group all developed tumors and succumbed to the disease. Based on the previous results, we demonstrated that tumor APCs can adopt CDC1 functions, they can activate T cells in vitro. But because we were always thinking about using CDC1 reprogramming as immunotherapeutic strategy, it was also important to address whether tumor APCs are actually able to induce the anti-tumor immunity in vivo. And to this end, we used mouse model, which was bearing B16 ova tumors. And as a therapy, we injected those mouse with reprogrammed B16 cells at which were stimulated with ovalbumin protein. And we did the injections at three subsequent time points at day 7, 10, and 13. And you can see that the administration of the tumor APCs managed to delay the tumor growth and also prolong the survival of the animals. And we validated this effect by showing that actually the intratumoral injections of APCs managed to expand T cells, which were specific not only against ova, but also against other endogenously present melanoma antigens, and both in peripheral blood and lymph nodes. Regarding the anti-cancer immunity, we proved that tumor APCs are working more on the side that they are effectively boosting the antigen presentation of tumor antigens, which then results in T cell activation. So from this perspective, it was we were very curious to see whether tumor APC strategy can actually work synergistically with immune checkpoint inhibitors, because these can then unleash the activation of T cells. So to test this, we decided to use the same mouse cancer model, and we combined the treatment with the intratumoral tumor APC injections with introduction of checkpoint inhibitors. And here we saw that especially the combination of the tumor APCs with checkpoint inhibitors slowed down significantly the tumor growth in the animals, and also significantly prolonged the survival of the animals, here shown as the green bar. And here on the right side, this is actually the first animal from our experiment in which the tumor receded completely and which basically survived as absolutely tumor-free. And you can see that it developed even this small depigmentation, which is truly associated with the response of the immune system against melanoma-associated antigens. So to conclude, I would like to say that we successfully showed that using PU1, IRF8, and BAT3, we can force CDC1 identity in cancer cells and generate so-called tumor antigen-presenting cells. Tumor antigen-presenting cells adopt multiple characteristics of natural CDC1 cells, and they are able to effectively present endogenous cancer-associated antigens and expand T cells both in vitro and in vivo, which leads to the reactivation of the immune system against cancer. We hope that using this approach, one day we will be able to overcome obstacles of current immunotherapy, which involve loss of antigen presentation very frequently or tumor heterogeneity, and that our data will help to develop a novel reprogramming-based platform for cancer immunotherapy. And in the end, I would like to thank to our group leader, Philip, also to Gabriela, Erwin, and Ilya, who did like a big share of this paper as well, and also to our collaborators from CCIT, University of Lausanne and Geneva, and many others. And thank you for your attention, and I'm very happy to take your questions.
All right, that was a great presentation. Thank you very much. We already have a couple of questions in the chat. I will start with the first one. So Mate asked, she said a beautiful talk. Do you think that the transformation of tumor cells to dendritic cells could spontaneously occur in tumors? For example, there are MHC2 expressing melanoma cell lines in numerous samples. Do you think that they resemble these induced tumor APCs?
This is hard to say, but we think that it's very specific to the reprogramming process, because in cancer cells sometimes you see expression of multiple markers that we also encountered, like several cancer lines or primary cells which are expressing like very low levels of MHC2 molecules. But I don't think that they would be by default, or that this like really the reprogramming into the CDC1-like cells would be happening like without introduction of those factors.
Right. Thank you. The next question from Di Chin is, thank you for the talk. is there any way to prove if the CD8 T-cell activation in vivo is induced by the direct presentation or cross-presentation of tumor-associated antigen?
Yes, we actually, I didn't mention it because of the time issues like in the presentation, but we also did experiments in BAT3 knockout mice, which actually don't have natural CDC1 cells. So we know that then when we see the same effect, same anti-tumor effect, that this needs to be elicited by the tumor APCs, because the natural CDC1s, which would kind of did the same effect, or the presentation of the antigens are not there. So we are sure it's like the effect we see is attributed to our tumor APCs and not to naturally occurring CDC1s.
All right. Thank you very much. Next question from Lejon. So cool work. Did you by any chance compare tumor cells lentivirally transduced with various co-stimulatory molecules to your reprogram cells?
Can you maybe repeat, because I'm not sure if I understand.
So if you compare not doing the full transformation of the disease, but just making the tumor cells express these co-stimulatory molecules versus the full transformation with all transcription factors into a dendritic cell.
You mean that if we only induce if we would like ectopically express the co-stimulatory molecules but didn't reprogram the cells?
Yes, I think that's correct, Lejon.
Right? No, this we didn't do, but since like a lot of the cells actually lose the expression even of the MHC1, I don't think they could efficiently activate when they activate the immunity if they would be only expressing the co-stimulatory molecules on top.